Now, yesterday we talked on mitochondria, the plastids or the chloroplast, and we have discussed also the similarities of the chloroplast and mitochondria in relation to the bacterial origin. So, one reason that the mitochondria and the bacteria are related because they behave like a bacteria in terms of replication, which is similar to the bacteria by binary fission. And at the same time, they have a genome, a circular genome. But the difference on the bacteria have singular circular genome, while the mitochondria is double-stranded. So double-stranded but circular. It is not linear. Linear, it could be found only on the nuclear DNA. Now, there's a question yesterday that why is it that there's no fraternal mitochondrial was passed on to the next generation? So the answer to that, during the fertilization of the egg, the, the axomere or the neck of the sperm was left outside the membrane of the egg and only the head entered the cytoplasm of the egg. So that is the reason that the paternal mitochondria was not inherited or was not included in the egg because it was placed outside together with the tail. So hindi na pass on. So that is the reason why only mitochondrial DNA was inherited from one generation to the other. So mitochondrial DNA was also considered as the maternal genome. So it derived from our mothers. So if we're going to test the relationship of our mother, mitochondrial DNA could be used as a tool to show the relationship between the offspring and the mother. And another thing that the mitochondria also accept other molecules like lipids and proteins. And in proteins, it passes to a certain complex. We have the TOM and the TIM23 complex. So these act as a pore for the passageway of the protein molecule passing through the outer mitochondrial membrane to the inner mitochondrial membrane, then to the matrix. Now let's have peroxisomes. Now peroxisomes also like a lysosome, but it is quite smaller compared to the lysosome. And it is one of the smallest organelle. Sometimes they are called microbodies. Now this was discovered by Rodin in 1954. Now, the name peroxisomes was derived because it is the site of synthesis of hydrogen peroxide. Now, hydrogen peroxide or yung agua oxinada is a metabolic waste product that needed to be broken down or transformed to another form because accumulation of hydrogen peroxide in a cell is very dangerous or fatal siya or poisonous. So it needs an enzyme, a catalase, to break down the bonds of hydrogen peroxide in order to be converted into water and oxygen. Now, the structure of peroxisomes, it is also like other organelle, it's double membrane bounded, and like a plus cell membrane, it has also some proteins, integral proteins, sa pa, phospholipids as a lipid component of the membrane. Like the mitochondria at the center, we have the matrix. And inside the matrix, you have the hydrolytic enzymes that will perform also metabolic and or catalytic function. So all the peroxisomes are morphologically similar to lysosomes in which they perform or engulfing particulates found in the cytoplasm. Like the lysosome, it will also act as an immune defense system or immune defense organelle to protect the cell from foreign material. Like the peroxisome, it also do the same thing sa lysosome, but it will take care on the smallest particle. Larger lang si lysosome versus peroxisome, smaller aspect. But the primary target is to break down the hydrogen peroxide as a result of metabolic byproduct. So they are assembled like mitochondria, chloroplast, and from proteins they are synthesized of free ribosomes, then imported into peroxisome as a complete polypeptide chain. So there's a similarities between the mitochondria and the chloroplast that peroxisomes also derive from protein. It is also um, accepting protein from the ER or the endoplasmic reticulum. And it also multiplies like the bacteria through binary fission. So peroxisomes do not contain their own genome. That is a difference between the mitochondria and the chloroplast, because chloroplast mitochondria has its own genome, but si peroxisome wala siyang genome. Now, what are the functions of the peroxisomes? Peroxisomes contains at least 50 enzymes. So 50 enzymes. Imagine that for a microbody, it carries a lot of enzymes. Peroxisomes originally were defined as an organelle but carry out oxidation reaction, leading to the production of hydrogen peroxide. But it also contains an enzyme that breaks the hydrogen peroxide. So let's have the fatty acid oxidation of peroxisomes. Like we have the fatty acid compound, then this will, as a byproduct of metabolic activity with the enzyme of the peroxisome, it has a byproduct of hydrogen peroxide. But since it contains an enzyme called catalase, it breaks down the hydrogen peroxide into two molecules of water and oxygen. So, hindi lang breaking down the hydrogen peroxide, but also it involves other metabolic processes. Now, peroxisomes involved in the synthesis. Yes, it also involves in the synthesis. It works with the mitochondria and other organelles. So, addition to providing compartment of oxidation reaction, it also performs again the oxidation reaction. So, it involves in the lipid synthesis. Now, in the diagram, we have the mitochondria and the peroxisome. Now, the fatty acid produced by the peroxisome will be accepted by the mitochondria. Then mitochondria, this fatty acid will be used in the TCA cycle or yung Krebs cycle to produce an energy. Now, it produces an ROS, or we call it the reaction oxygen species. So there's an oxygen will be produced. Then this will be used by the in the redox potential reaction. Now, in peroxisomes, they do the anabolic reaction in which the fatty acid converts into acetyl-CoA. And since acetyl-CoA could be also used in PCA. So this will help the mitochondria to produce a lot of energy because the peroxisome helps in producing acetyl-CoA. So hindi lang other organelles or hindi lang ang yung glycolysis in the cytoplasm helps the mitochondria to produce energy, but also si peroxisomes. So aside from anabolic reaction or metabolism, one primary function ng peroxisome which is known to all because kaning iyahang function on metabolic lipid synthesis was not mentioned in other references, but it primarily in most regular general references, ang function lang ni peroxisomes is to break down hydrogen peroxide. Yun lang ang nakalagay sa ibang references. But in higher cellular molecular biology books, dito na na-highlight ang function ni peroxisomes. So peroxisomes also involves in the development of plasmalogen. So in the liver, peroxisomes are also involved in the synthesis of bile acids. And we know what is the function of the bile acids in our digestion. So which derive from cholesterol. So it converts cholesterol for the help in the synthesis of bile acids. And also it contains enzyme that requires the synthesis of plasmalogens. So plasmalogens is a member of the phospholipid family, which is important for the membrane component of some tissues like in the heart and in the brain. So plasmalogen could not be found in any other areas but could only be found sa heart and the brain. So very important ang function ni peroxisome. Aside from that, peroxisome also, especially in plants, so sa plants naman, ang role ni peroxisomes, it involves or responsible for the conversion of stored fatty acids to carbohydrates. So it converts fatty acids to carbohydrates and to be used in a glycosylate cycle. Now the glyoxylate is a cycle also that resembles the citric acid cycle. So pareho lang siya ng process. So this is the glyoxylate cycle which resemble the TCA. So ano lang ang meron sa glycosylate cycle? Say glycosylate cycle don't have isocitrate and ketoglutarate. So the citrate will be transformed directly to succinate or glyoxylate. So hindi na siya mag-transform into ketoglutarate or isocitrate. Now in TCA, sa TCA naman, the isocitrate will not transform into glyoxylate, but it will transform to ketoglutarate and succinyl-CoA, producing 2 molecules of carbon dioxide. Yun lang ang sa TCA. But in this cycle, this reaction is not present. So kaning naka-highlight na faded, this is for TCA. And kaning darkened fonts in the cycle, ito yung cycle in the glyoxylate cycle. So parang pareho or 80% similar to TCA, but only difference is the isocitrate will not converted into ketoglutarate to succinyl-CoA to transform to succinate. It will directly transform to succinate or glyxooxylate. Then the glyxosylate will transform directly to malate. Pwede din siya hindi mag-develop or transform into fumarate. So peroxisomes in which takes place are sometimes called glycosomes. So that is happens sa plants. Now the second, peroxisomes in leaves are involved in photorespiration. Now photorespiration serve to metabolize a side of product formed during photosynthesis. So this is very important in photosynthesis. Now while the chloroplast having the Calvin cycle on its matrix, it releases phosphoglycerate or photoglycolate. then transform into glycolate. Now, since glycolate is a harmful substance that needed to be converted into a glycine. So that will be the help of, or the peroxisome will do that. So it transforms the glycolate into glycine. Then the glycine will be used by the mitochondria in its cycle to produce carbon dioxide, ammonia, and serine. Then the serine will go back and will be used again sa peroxisome to transform to glycerate. And now, the glycerate, when attached with the phosphate group, it turns into phosphoglycerate, which is very important in the Calvin cycle. So that is the importance of peroxisomes on the leaves of a plant. So the Calvin cycle, it says that this 2-carbon glyceraldehyde phosphate is needed to be transformed kasi mag-transform siya into glycolate. So, yun yung pinakita sa performance ni peroxisomes. Bakit kailangan siya sa plants. And sa other references, si peroxisomes was not mentioned as the difference between the animal cell and the plant cell. It said that peroxisomes could only be found sa animal cell. But now we found out na peroxisomes are present in a plant cell and it performs metabolic action transforming the glycolate into a usable compound called the glycine, and the glycine will be used the mitochondria to transform it into serine, and serine transform back to glycerate in order to be involved in Calvin cycle. So, kung absent yan, then it will be dangerous for the plant. Saan naman, hindi naman mag-transform. Okay. So it is converted, the glycolate, and then the transfer to peroxisomes where the oxidized and converted back to glycine. So ito yung sinabi ko kanina. Nauna ang sinabi ko na ulahi ang PowerPoint. So it is first converted to glycolate again. Then si peroxisome transformed into glycine. From glycolate to glyoxylate, then glycine, then give it back to mitochondria to convert glycine to serine. Then the serine will be transported back to the peroxisome to convert it again into glycerate. Then the glycerate will be added with a phosphate group to become phosphoglycerate. So 2 phosphate group will be added in the phosphoglycerate. Kaya siya 2, 3. So nasa position 2 and 3 na carbon na attach ang phosphate group. Okay. So peroxisomes play an important role by allowing most of the carbon in glycolate to be recovered and utilized. So converting the harmful substances and utilized to be useful again. Now, how about the peroxisome assembly? Now, peroxisome receive proteins from the cytoplasm. After the ribosome synthesizes or translates the protein, then it will be imported to the peroxisome. Now, the peroxisome become a complete polypeptide chain, will be directly absorbed by the peroxisome. Now, the peroxisome will divide. So, This will help them develop new organelle by binary fission. So this will enable the peroxisome to grow. So by binary fission. So that's all. Yun lang kasimple ang peroxisome.